All right, so good morning. So it's Monday, August 31st, the last day of the summer. Crazy times, but we'll get through this. We'll get through this, and we're doing everything possible to stay in a game. So we are doing online teaching. Not easy, but we're gonna stay on the path to knowledge. All right. So we are going to be working on chapter 3. We're going to start chapter 3 today. Proteins, amino acids, peptides. So as usual, so I'm recording this meeting. I'm going to put it on YouTube after that. So if you want to listen to it again for any reason, again, I encourage you to take notes as we speak, right? And encourage you to ask questions. Ask questions so that I know that you're there and I know that you're listening to me and I'm not speaking into my computer and see some of you. I can see some of you, but most of you have just your names. Just your names. But I'm going to minimize the icons anyway so that I can see the slides. All right, so let's go to the slides. Let's start sharing. Now, any questions before we start about anything in this class? I do have a request. I want more participation in the discussion on Canvas. Let's learn all together. All right, so I'll minimize this. All right, can you see my slides? The first slide?
Yes.
Learning goals. All right, remember, we're gonna start with our learning goals, learning objectives. And well, there are 20 amino acids, right? So you know that proteins are the most abundant molecules in biological systems, right? Have lots of functions, lots of structures, and all that is based on 20 amino acids. So to understand the properties of proteins, to understand the properties of the most useful and most abundant biological molecule, we need to know the structures of its constituents. And there are 20 of them. Well, there are more, but we're going to focus on 20 because these are the most abundant ones. We're going to learn their structures. We're going to learn how to name them. And based on their structures, we're going to learn their properties. So that should be your number one goal in this lecture, is to get this material into our brains. Then obviously these amino acids by themselves, some of them are useful by themselves, but most of them are only useful as part of peptides. Now peptides are small proteins, so we're going to learn about those. Okay. We're going to learn about ionization behavior, very important. Now many of these amino acids have carboxylic acids side chains. Many of them have basic side chains. And those will dictate the properties of the proteins because the proteins will be charged. And some of them will be protonated, some of them not protonated. And remember, in the previous chapter, there was a reason why we spent well, not two lectures, but one whole lecture on water. And we learned, I spent quite a bit of time talking about hydrogen bonding. Hydrogen bonding, that's what proteins are based on. All their interactions with other molecules, all their three-dimensional structures are based on hydrogen bonding. And so hydrogen bonding depends on ionization behavior of these amino acids and peptides. And then once we get that done, and then we will move on to methods to characterize proteins, peptides and proteins. Again, the way to study for you guys is to focus on these 4 concepts and kind of group the material under these 4 concepts and expect to be tested under the umbrella of these 4 concepts. All right, so what do proteins do? Just a brief overview. Catalysis, right? So the many proteins are enzymes. So for example, enolase, right? So let me,
No.
did you want to say something? Somebody want to say something? Yeah? No? Okay. So if you don't want to say anything, just mute yourself, please. Okay. And if you speak, please do not forget to unmute yourself so we can hear you. All right. So for example, enolase. So there's an enzyme in glycolytic pathway, right? So glycolytic pathway. Anybody knows what this pathway is? It actually kind of the names tells us what this pathway is. What do you think glycolytic pathway is?
Or glycolysis?
Glycolysis, that's right. Glycolysis, energy harvesting pathway, degrading food, right? Now, DNA polymerase is an enzyme responsible in DNA replication, right? So it'll double our DNA when a cell undergoes cell division. Another important function for proteins is transport. Hemoglobin in our blood, right, transports oxygen in the blood to the tissues. Oxygen just doesn't, it dissolves very poorly in water, as we learned. It's a non-polar molecule, right? Doesn't dissolve in water well, so we need some special transport system, and hemoglobin is part of that. Now, for example, lactose permease, right? So lactose is sugar, allows for lactose to get across cell membrane. Structure, collagen, connective tissue, keratin, hair, nails, feathers, horns. Motion, right? So muscle tissue, so very important proteins, myosin and actin, cell motility, right? So actin is very important in cell motility. So remember, for a cell to move, it needs to be able to change its shape, right? So just imagine a cell sitting in a 3-dimensional matrix surrounded by other cells and surrounded by extracellular matrix. How will it get out of there? You know, it's stuck. It's stuck, right? It needs to change shape. It needs to be able to squeeze in between other cells and get out of there. And obviously, one thing that comes to mind is cancer, right? Cancer metastasis. How do cancer cells metastasize? So obviously, we know that cancer by itself, if it was localized, it probably is not such a big killer. It'll just sit there until we can surgically remove it, and there should be no consequences. But it's actually metastatic behavior of cancer cells which kills us. In fact, 90% of cancer patients die from cancer metastases, not from cancer itself, right? And so, and how do cancer cells metastasize? Well, they have to be able to escape the tumor tissue, right? They have to be able to escape the tumor tissue, get into the bloodstream, get carried by the blood to another tissue, get out of the bloodstream, and start a new growth somewhere else. And so actin will be key molecule, key protein responsible for the behavior of cancer cells. And so actin, no wonder that actin is actually a big target in treating cancer. Unfortunately, drugs which target actin are highly toxic. Now, what did I want to say about rhinoceros? Black rhinoceros, it's horn, aphrodisiac, right? But it's a protein. It's a protein. Let's see. It's a protein.
Aren't rhinoceros horns just keratin? It's basically just modified hair.
Perfect, keratin. So it's a keratin, and so keep in mind that keratin, the same keratin as in our nails, right? Same as in our nails. And so, for example, there's a black rhino. Part of the reason why it's getting extinct is because there is belief among many poachers. Well, not poachers. Poachers do their job, but many people who buy these horns from the poachers believe that these horns have aphrodisiac properties. But if you just take this horn, if you grind it into a powder, then you clip your fingernails, grind those into a powder, you'll end up with exactly the same protein. It's exactly the same protein, keratin, as you said. Perfectly noted, yes. So who would have thought? Yeah, very strong protein. All right, what else? All right, so proteins, heteropolymers, remember what polymers are? Polymers are large molecules which are composed of monomers. Remember monomers? Monomers polymerize into polymers. And in most cases, polymers are composed of the same kind of monomers, but proteins are hetero. Let me actually turn this into a pen. Hetero is a very important prefix, which tells us that it's not the same amino acid, right? If it was, let's say, glycine, polyglycine, that would be a homopolymer, would be just a homopolymer. But since we're dealing with 20 amino acids and they're all different in each protein, we're dealing with heteropolymers. Right, so amino acids have properties. And so why amino acids? Why are proteins composed of amino acids? Well, because they have the capacity to polymerize, right? You need to have a monomer that can undergo polymerization. You need to have a monomer that once polymerized, it will have useful acid and base properties. Remember hydrogen bonding. We want proteins to be able to make those. Varied physical properties, right? We want keratin in the rhino, in the black rhino. We want this keratin to be very strong. And there's a reason why keratin is very strong, right? Because it's physical properties. And varied chemical functionality, right? So enzymes carry out lots of reactions, and the reactions that enzymes carry out are based on the chemical functionality which is present at the active site of these enzymes. All right, so here's how amino acid would look, right? So what do we have? We have an alpha carbon. Now remember, in any amino acid, so let's say if I'm gonna draw amino acid, you will have a carboxylic acid, just like that. You will have an amino group, like that. You will have a carbon. And then it's possible that you will have something attached here, okay? So this carbon here, so you can number things in amino acids. So this will be carbon 1. This will be carbon 2. This will be carbon 3, and so on and so forth. Alternatively, alternative system is to call this carbon alpha. This will be carbon beta. Then you have gamma, right? Gamma, delta, epsilon, and on and on and on and on. Okay. So alpha carbon. In most amino acids, this alpha carbon, in most but one. Let's see if you have some prior knowledge, would be good. So in most amino acids, this carbon is tetrahedral. So in other words, it's asymmetric, right? So it has 4 different substituents. It have a carboxyl group, has an amino group, has a side chain, and has a hydrogen. In which amino acid is this carbon not chiral?
Glycine
Perfect. And why?
has a hydrogen instead of a carbon, another hydrogen.
That's right. In glycine, takes me a while to write. In glycine, R equals H, which leads to the fact that C alpha is not asymmetric. No, asymmetric just one S, right? Ah. What did I just do? Did you have a chance to take a note of that? Okay. Sorry, my writing.
Control C, undo that.
How can I undo that?
Control Z, maybe.
No.
You can try Command Z.
Yeah, I'm trying Command Z. It's not doing it. All right. Anyway, you have this in the textbook anyway. So, but you got the most important point here was the idea, right? So you realize that the reason why it's asymmetric is because there's a side chain, and glycine, the side chain is hydrogen. So the carbon is not asymmetric.
I have a quick question about this. In the body's pH, is the carboxylic acid, is that usually deprotonated and the amine is usually protonated? Or
Yeah.
how does that work for those two?
Yeah, good question. We'll talk much more about those and those kind of questions. But in general, so if we need to, we spent a little bit of time talking about pH and pKa, and remember pH and pKa. So I'm going to write something else here, and I hope I will not erase it this time. When, now this is very important for understanding of behavior of amino acids and proteins. When pKa, no, sorry, when pH, let's do it this way, equals pKa over carboxylic acid, %, is deprotonated. Deprotonated.
So that's when it is a carboxylic acid.
Yeah, so HA Here's your carboxylic acid. HA goes back into H plus, back and forth between H plus and A minus. Okay, so 50% of this, the ratio is going to be 1 to 1. Okay, now you can manipulate this. So this equation here. So let's say pH, let's say the pH is now lower than the pKa. So let's say the pKa is 4, and let's say pH is 4, then you get 50% of each, right? But let's say you put this in an environment where pH is 2. Will your acid be more protonated or less protonated? So in other words, where will the equilibrium lie as you lower the pH?
More protonated?
More protonated. Remember, what's the principle? Le Chatelier principle. So as you drop the pH, you increase the amount of the protons, right? It becomes more acidic. As you increase the amount of protons, the system responds by removing the protons and moving to the left to give you more protonated acid, to give you more acid, right? The reverse is true if you raise the pH. Now, let's say your pKa is 4 and you raise the pH to 7 by 3 units, then basically you will start removing the protons from the amount of protons is small because you raise the pH, right? And then more of the acid will undergo deprotonation to give you protons. So you can manipulate this by changing the pH. And the key here is to know the pKa. So you need to know the pKa of carboxylic acid. Sorry, not carboxylic acid. You need to know the pKa of the, yes, carboxylic acid. You need to know the pKa of the ammonium. And then you will know which one will be protonated and which one will be deprotonated, right? So in most cases, well, actually, we will look at a specific example, but this is a key slide. Keep coming back to this. Don't erase. No, I will try not to erase it this time. But pH equals pKa 1 to 1. That's the key, because this is where you start with. This is where you start with. You lower the pH, you go left. You raise the pH, you go right. And the protonation, deprotonation will be dependent on the pH. On the pH value. Okay. This makes sense? All right, so we will develop this concept as we go further into the lecture. All right, so alpha carbon, always tetrahedral, except for proline. We'll look at the structure of the proline. Basic connected to hydrogen, force substituting is unique in glycine. It's the simplest amino acid. The force substituent is also hydrogen. So we just talked about that. All right. I'm going to actually come back to this. Yeah, maybe let's do this now. So, out of 20 amino acids in our proteins, all 20 amino acids are L amino acids. That's the key, not D amino acids. And so what does this L and D actually mean? Well, it can be R, it can be S. We don't know. Okay, it's not R and S. We're going to go by L. And so what L means is that if you draw the amino acid in this way, so put the carboxylate on top, carboxylate on top, put the amino group on the left, and if you end up with your R group on the bottom, R group on the bottom, and your hydrogen on the right, then this is an L amino acid. Now, if you keep your carboxylate on top and R group on the bottom, but the hydrogen and amino group in the amino group switch, but their positions, then you go to D amino acids. So L amino acids are all found in our 20 amino acids. Now, D amino acids are also found in some organisms, particularly in the microbiological world, right? So for example, in the cell wall of bacteria, for example, it's crucially involved in the mechanism of penicillin. Penicillin works by breaking the bacterial cell wall or inhibiting the biosynthesis of bacterial cell wall. We just talked about osmosis when we talked about water, right? And I told you that osmosis kills bacteria very easily, and bacteria resist because the cell can shrink, or this, if it's hypertonic, or the cell can burst if it's hypotonic environment. But the bacteria build a cell wall around it to resist these osmotic pressures. And one of the key components in a cell wall is D-alanine. D-alanine and penicillin actually interacts with the construction of the bacterial cell wall by actually interacting with a portion of the cell wall that has D-alanine in it. So can make a note that it's important for penicillin penicillin mode of action. But we're going to be dealing with L amino acids, and the question is why. And the reason is in 1891 was a chemist, Emil Fischer, who basically knew that glyceraldehyde that has this structure, glyceraldehyde, remember these are Fischer structures from OChem1, right? Fischer projections, OH, H, and you get that. So this is L glyceraldehyde. L-glyceraldehyde. And you can see here, the OH is on the left, H is on the right. And by analogy, L-alanine will have amino group on the left and this on the right. And so originally, remember, L and D was L levorotatory, D dextrorotatory. So L in the will rotate the plane polarized light in the left direction, D in the right direction, right? And this was the structure that was assigned. And so the Fischer proposed to name amino acids after the same phenomenon, after L-glyceraldehyde and assign all these 20 amino acids an L configuration. So L-alanine.
Oh, I have a question. Can you hear me?
Yeah, I can hear you.
Okay. Yeah, so just to like recap, if the alpha hydrogen is on the right side, that would be L, like in the nomenclature, right?
Yeah,
Like L, whatever. Okay. Thanks. Thank you.
yeah. Well, the amino group on the left, that's what you look for. L, left, amino group. Yeah. Okay, so we will look at the structures in a moment. For now, I'm gonna jump to this slide and talk about the names of these 20 amino acids. Very interesting actually where the names came from. So for the 6 amino acids, so let's do C, which is cysteine. You can find cysteine here, right? Cysteine. H, can you find H? What's H?
Histidine.
Histidine. Now, can you find I? What's I?
Isoleucine,
Isoleucine. Can you find M?
methionine,
Methionine. S.
serine,
Serine. And V?
Valine.
Yeah, so valine, yeah. So these are 6 amino acids which are unique. In other words, no other amino acid starts with the same letter. So this was an easy job of naming these ones. Now you may notice, so now for each amino acid, so we have a full name, right? Then we have a 3-letter name, which is in all cases, if I'm correct, all cases, well, almost all cases, is the first 3 letters of the full name, which is correct for glycine, valine, leucine. Well, isoleucine, not exactly, right? So there's some difference here. Tryptophan is also, there's some, not exactly, but this is still correct. Asparagine, not exactly. Glutamine, not exactly. Lysine, histidine, arginine, aspartate, glutamate. Okay. And then there is so you would think that, why do we need so many names for one amino acid? Well, for a growing area of bioinformatics, required even shorter names, right? And so especially for computing purposes, right? So a lot of protein work these days is done on computers, and we have models actually to predict how various proteins going to behave. Even we have ways to predict their 3-dimensional structures on computers now. You given a 2,000 amino acid long protein, you can put it in a computer, and the computer can actually pretty well efficiently tell you what it's going to look like in 3 dimensions once the protein folds. Yeah, protein is not going to be just a long ribbon like a snake, all but well in its stretched shape, right? So it's all going to be wiggled and entangled and forms various folds, loops, and so on and so forth. All right, so these 6 are unique. Now, Now for 5 others, we're gonna assign the letter based on, give it to amino acid which is the most common in proteins. For example, leucine, right? We give L, even though lysine is also L, the first letter is L. But we're gonna give L to leucine because leucine is much more common in proteins. So this will be A, alanine. This will be G, glycine. L, leucine. P, proline. And T, threonine. Okay, so most common in proteins. All right. For another 4, we're gonna use something very interesting. We're gonna use a letter which is phonetically suggestive. So what does that mean? So for example, R. What's R? Can you find R?
Arginine.
Arginine. So R. Arginine. Arginine. How about F? What do you think F is? Well, you can find it, obviously. What's F? Can you find F?
Phenylalanine, if I'm saying that right.
Yeah. You can say it many different ways. Phenylalanine. Don't worry about pronunciation. Most people will know what you're talking about because they don't know how to pronounce things themselves anyway. For example, I mean, especially for me, when with a foreign first language, but for example, you know, I got my PhD in Boston at Tufts University, and there a functional group amide, like amine. If you attach a carboxylic acid to it, it's amide. Here it's amide in Texas. Amide versus amide. Here it's nitrile. In Boston, it's nitrile. So yeah. Now, why? What's why?
Is it tyrosine?
Tyrosine. 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 And there is one more, which is the funniest of all. W. Can you find W?
Tryptophan.
So why is tryptophan W? Who actually put this on the test? Just to make sure you're paying attention in class. Why is tryptophan a W? Because some people, like my little son who is 5 years old, cannot pronounce the letter R very well. He would say tryptophan. Maybe the person who couldn't pronounce the R, who could not roll his R, he would say tryptophan. And he gave it the letter of W. Tryptophan. I think the origin of these is very interesting because, well, the reason why I'm giving this to you is not just for curiosity. I hope this will help us, me too, because actually every time I teach this class, I keep forgetting some of the one-letter abbreviations. But hopefully these origins of these single-letter abbreviations will help us remember them. And then the rest were harder to assign. So there are 4 left. So there's D. D would be aspartic acid. Then N. You might have found N, right? That's asparagine. Where do you think asparagine was discovered?
Asparagus.
Asparagus, correct. Where do you think tyrosine was discovered? Who knows the French word for cheese?
Fromage.
Fromage.
Wasn't it like,
It is fromage. Why am I thinking
I think it might be the Greek word, tyros or something?
Greek. French,
Yeah.
fromage. Thank you very much. At some point I knew French a little better than now, but because I worked in Montreal for a while and I learned some French, but I forgot the fromage. Yeah. Okay, asparagine. Asparagine. And here where the N is. Oh wait, why do I have, oh, I don't wanna use the eraser anymore. I'm just gonna do this. Asparagine. And then what else do we have left? We have glutamate, which is E. Glutamic. Yeah, I don't know really. This one is really questionable. Glutamate. And the last one is, no, the second to last one is glutamine. And that will be cutamine. Cutamine. And what's the last one? Which one haven't we done yet?
Lysine, K.
Yes, lysine is what, and why is lysine K? What do you think? Well, L is already taken by leucine, so no luck there. So why is lysine K?
Is it like vitamin K?
Nah.
Is it just that K is the letter before L?
Yep. When you were little, you knew this song: A, B, C, D, E, F, G, H, I, J, K, L, M, N, O, P, right? So K is close. close to L. All right. I think that's enough for this exercise. So where are we in terms of time? Okay, we still have more than half an hour.
Is there a reason that those ones are grouped like that, or is it just they were the ones that got kind of left out and were the harder ones to find like a one letter abbreviations for?
Yeah, I guess so. Yeah, I guess the first 6 were the easiest ones because they were unique, right? Then they went by the most common one. Then the ones that were not unique, right? Like for example, alanine and aspartate, they went by the most common one, or glycine or leucine, proline, threonine, as opposed to tyrosine. And then, yeah, and then the rest, you kind of just have to be creative.
Okay, thank you.
All right, so let's look now at the structures. Let's look at the structures. So even though there are 20 of them, but hopefully we will group them into categories in our mind, not on the basis of the name. The name, as you can see, doesn't really tell us much in terms of what this amino acid will do once it gets incorporated into a protein. What will tell us what it will do is its properties. And so here we're going to actually put all amino acids into 5 major groups, right? So again, it's all about polarity, ionization potential, all the properties we learned in organic chemistry. Guys, organic chemistry is the main foundation for biochemistry, right? If the reaction does not exist in organic chemistry, it will not exist in biochemical system, and vice versa. So all chemical processes are identical, they only occur in different environments. So I strongly encourage you to, if you have taken your organic chemistry a while ago, really keep your organic chemistry textbook and lecture notes by your side, and review the material. Keep reviewing the material. Plus, everything is online these days, right? If you forgot what aliphatic is, just look it up, Google it, and you'll find it right away, right? So there will be 7 nonpolar aliphatic groups, 3 aromatic ones. There will be 5 polar but not charged, right? So they'll have a dipole moment, but they will not be charged. And then there'll be 3 which are positively charged. And remember, the reason why they will be positively charged. Okay, let's, before we even go into details, why would something be charged, which probably means protonated, right? Why would something be charged at physiological pH of 7? What would be the pKa of such charged species? What do you think? So if something will remain protonated at pH 7, what should be the pKa be?
Should be higher than 7.
Be higher than 7, that's right. And so, for example, amino groups, so we will see the amino acids with nitrogens in them, right? So those, once they protonated, they have pKa's 9 and 10. And so that's why they will be positively charged. And negatively charged is vice versa, right? So if the physiological pH is 7, so the pKa and probably carboxylic acids, pKa's are around 4 and 5. So those will be deprotonated, right? So keep in mind physiological pH of 7, and then you go by the pKa. If it's above it, protonated. Below it, deprotonated. All right. So these are nonpolar aliphatic R groups. So we talked about glycine. So nothing, no R. The R group is the hydrogen. This is the simplest of all amino acids, the smallest of all amino acids. You have carboxyl group, amino group, and just one carbon in between. Easiest to remember, glycine. Alanine is the next one in complexity. So we replace the hydrogen with the methyl group, but still pretty simple amino acid. Now, proline is very different from all the other ones because it's the only amino acid in which the nitrogen, the amino group, is incorporated into the ring. So let's see what's happening here. So you have the R group coming off the alpha carbon. This is the alpha carbon, right? The R group coming off the alpha carbon, going this way and forming a bond with the nitrogen, kind of twists around and forms a bond to form a ring, right? So that's the only amino acid that does this. Its name is proline. And you can already even predict some properties of proline. What do you think, what's the main property you think that will be affected? Compared with all the other amino acids, like what would proline be famous for? How about flexibility? Will it be more flexible or less flexible than the other ones?
Probably less
Yes.
flexible because of the ring.
Yes. When you confine something to a ring, you basically lock it into a ring. Lock it into a ring, and so any protein that has proline in it will be conformationally less flexible, right? It will not be able to rotate freely around its single bonds. All right. Next one is valine, right? So here the R group is starting to grow, getting bigger. Now we have isopropyl group here. There's leucine. Leucine, the difference between valine and leucine is we're going to insert a CH2 group in between. Isoleucine, the difference is that the methyl group will migrate to this carbon, and that's why it's called isoleucine, because it stands for isomeric leucine, right? So it's an isomer of leucine. And methionine. Now, methionine, it has an electronegative element, so you would think that it should be should not belong in this group because it has a dipole moment, right? But what do we know about sulfur? How electronegative is it? So remember periodic table. Remember where sulfur is? Can you, without looking at periodic table, can you remember where sulfur was?
Is it right below oxygen?
Below oxygen. So what happens with electronegativity as you go down in the periodic table?
It lessens.
Lessens. So which means that the difference in electronegativity between sulfur and carbon is not as pronounced as it is between carbon and oxygen. So for that reason, methionine is not very polar, right? Because simply sulfur is not electronegative enough to pull electrons away from the carbon. All right. Now, next group, aromatic. So there are three aromatic amino acids. One is the simplest, phenylalanine, right? So this is by alanine, to which we just attach the phenyl group. So phenylalanine. Now tyrosine, the cheese in Latin, tyros, right? We were just told. So this one, the difference is there's an OH group, right? So for example, if you proteins which want some hydrophobic interactions, they will incorporate a lot of phenylalanine in them because benzene rings are very hydrophobic. But if you want some hydrogen, to preserve some hydrogen bonding capability, then instead of phenylalanine, you will incorporate tyrosine, right? So, so the proteins will use these judiciously. And then there's a tryptophan. Very interesting amino acid. Anybody remembers the name of this heterocycle? Remember, heterocycle is a cycle in which one or more carbons are replaced by heteroatom, such as nitrogen or oxygen or sulfur. Anybody remembers the name of this one? This ring here? Okay, this ring is called indole. It's capable of some beautiful chemistry. And you can see the surface area of indole is significantly larger than that of phenylalanine or tyrosine. So you would use indole in areas where you need you have a flat hydrophobic pocket that will be occupied by this. And it's also capable of hydrogen bonding. It has an NH. So these are aromatic R groups.
I'm sorry to interrupt. You said the ring on the tryptophan, the indole is hydrophobic. Is that correct?
Yeah. Mm-hmm.
Okay. Thank you.
It's hydrophobic, but it's still capable of one hydrogen bond, right?
Yes. Thank you very much.
Will we be tested over these R groups?
Sorry, repeat it.
Like, will we be tested over the structures of these R groups?
Yeah, you need to know the amino acids. You need to know your amino acids. It'll take you a while, but that's why I was spending quite a bit of time, and that's why I'm trying to give you as much information as possible, because it's not just a question of memorizing your alphabet where there's a Y is L after K. Yeah, Y is M after L. There's no primary or reason for that. So here, everything is logical. Everything kind of makes sense. So pay attention to, take notes of what I'm telling you, read the textbook, and don't just blindly memorize these structures. Don't just blindly memorize the abbreviations. Go by all these explanations, go by all the properties, right? And in most cases, so my approach to memory, my approach to learning material, that is a little sort of kind of a catalog, kind of tabulated more rather than conceptual, right? Is actually learning the properties, learning as much information as possible about each component. And then the structures and the names, they will just stick, yeah? And doing problems, you know, the work on sapling problems. Now the problems in the text, right? So let's see. Problems in the text. The sapling problems. Sure, you can find tons of problems online on biochemistry, different textbooks, solved, non-solved. So if you have time, obviously, this is not required, but that's how I would learn my amino acids, is by actually by learning everything I can about my amino acids, and then the structures, they will just stick. Now I have to say also that many proteins absorb in the UV region of light, okay? And that's primarily due to these aromatic amino acids. So tryptophan and tyrosine, they absorbed around, I think, 280. Yeah, tryptophan 280 nanometers, and tyrosine is just 276 nanometers. So in this region, aromatic amino acids absorbed, and for that reason, proteins absorb UV light. And you can actually study protein function and protein behavior by through their absorption. So polar uncharged R groups. So now you can see these have polar groups. These have polar groups, lots of dipole moment. Now we have a serine, right? So it's capable of hydrogen bonding, and it's electronegative. Threonine, same thing. The only difference is this methyl group here. So serine and threonine are usually two amino acids which can be found in the same area because they can perform very similar functions. The only thing is threonine is slightly larger because of the methyl group, right? So it can occupy an additional hydrophobic pocket in a protein. Now cysteine now belongs to this polar uncharged group, and primarily is because of this SH. So SH is capable of hydrogen bonding. So even though again, sulfur is not as electronegative as oxygen, but we're gonna put it in here because of the hydrogen bonding capability. Asparagine and glutamine. So these two are polar because of the amides. And remember, in amides, you have this lone pair on this nitrogen, which can move over, and the electrons can be picked up by this oxygen. So you get partial negative charge on the oxygen and partial positive charge on the nitrogen. So there's quite a bit of dipole moment with the amides. So that's why they're polar, and these side chain can, and they can also form hydrogen bonds, right? So there are two hydrogens here. This oxygen has two lone pairs. So there's a lot of hydrogen bonding capability. And finally,
Oh, I'm sorry.
Yeah.
I have a real quick question about the UV light absorbing thing. So can only proteins with aromatic groups do that, or you said all proteins can absorb UV light?
well, most proteins will have aromatic groups. So it's
Okay.
very unusual for a protein not to have an aromatic group, right?
Okay.
So
But like glycine or something would still do that?
if you have polyglycine, no, that will not absorb UV light. No. But
Okay.
but polyglycine, it's not really a biologically important protein, right?
Okay. Yeah, I
So,
see what you're saying.
yeah. So most proteins will have some percentage of aromatic amino acids in them.
Okay. That makes sense. Thank you.
Yeah, and they will absorb due to that. All right, and now charged. So we have positively charged R groups and negatively charged R groups. So here's lysine. Remember the abbreviation K because it's right next to L. Here's lysine. So it has a long hydrocarbon chain and a positively charged NH3 on the end. Again, it's positively charged because the pKa of this is around 9, right? Which is above the physiological pH. If I raise my pH, above 10, then I will lose this proton. I will lose this proton because the pH is now above pKa, and the solution is too basic for this nitrogen to hold on to the proton. It will become deprotonated. Then arginine, so something that you may not have seen type of group in arginine. So this type of group here is known as guanidine. Guanidine. So you can draw resonance structures here, right? So which one do you think is more, now both are protonated, right? So lysine and arginine are protonated. Which one do you think is more basic? Like which one will hold on to its proton more strongly? Lysine or arginine? Remember, the ability to hold on to the proton depends on the ability to stabilize this positive charge.
The arginine?
Yeah, why do you say so?
Because there's a second nitrogen that the charge could
Yep, that's right.
bounce around to.
In fact, there are two of them, right? There's this nitrogen and there's this nitrogen. And in fact, something for you to practice at home, draw a couple of resonance structures. So here, these electrons, oops, wrong. Okay, let me try. Erase a pen or erase?
Eraser.
Eraser. Yes, thank you. All right. The pen. So we can form a double bond here. And these electrons will move over to this nitrogen, right? And you can draw the resonance structure for that. And you can do the same thing with this nitrogen, right? So guanidine group on the arginine can stabilize the positive charge much better than lysine, right? So arginine without the proton will be much more basic and it will be protonated more easily at much higher pH values. It will hold on to the proton more strongly. And the last one, as far as the positively charged ones, is histidine. Now, so this 5-member tring is known as imidazole. Yeah, take notes, guys, because I don't know if actually they cover this in a text. This is imidazole. This is imidazole. And so in the imidazole, let me actually ask you something. Okay, so this is the imidazole. And now tell me, once this undergoes protonation, right? In fact, for some reason, there's a mistake in this slide because this imidazole is not protonated. When we protonate this, what site in the molecule will undergo protonation? Obviously, one of the nitrogens, right? But tell me, which nitrogen? The top one or the bottom one?
The bottom one.
So why not the top one? What's the problem with the top one?
Is it because of the double bond?
The double bond, there's one double bond here, one double bond there. What about the double bond? There's something else. Now, when you protonate something, look for a lone pair of electrons that you're going to protonate, right? So the proton is going to sit on a lone pair. That's where it's going to go. Maybe I'll eraser. I'll make this slightly clearer. Now, so in your notes, why don't you draw the lone pairs on both nitrogens and see which one will undergo protonation? It's very important. It matters. It's not that both of them will undergo protonation. Only one of them will. And that's very important for the biological properties of this amino acid, histidine.
Isn't the one on the bottom going to be the one that's protonated, because the one on the top, those electrons are contributing to the aromatic system?
Perfect. Yep, that's right. So these two electrons right here are part of the aromatic system, like that. Yeah. And these two electrons are actually perpendicular to the aromatic system. They actually align in the plane of the screen, right? They cannot be part of the aromatic system because they're not part of the p orbitals of the aromatic system are perpendicular to this lone pair here, right? And so this lone pair will grab the proton. The lone pair will grab the proton. But then keep in mind, you can also draw resonance structures, right? You can draw resonance structures. This can go this way. And this can go this way. So, and finally, we have two carboxylic acid containing R groups, and those will be deprotonated at physiological pH because their pKa's are around 4 and 5. Actually, it will be even smaller, maybe, being next to the nitrogen. Nitrogen pulls electrons away, which will make these more stable, right? Remember, to make a negative charge more stable, you have to pull electrons away from it. And so that's what this nitrogen can do, and which will allow for these to undergo deprotonation more easily. And so the pKa may be even lower than 4, could be around 3, 2.5. And so this is aspartate, and the difference between aspartate and glutamate is just the presence of one CH2 group. Aspartate isolated from, no, asparagine is from asparagus. I don't know where aspartate is isolated from. All right, so we had that. And I think we're getting close to the end of our time. We will briefly talk about unnatural amino acids on Wednesday, and move on to finally talking about what the question was about earlier about the ionization of amino acids, right? So basically very important for the properties of proteins. Any questions about anything that was said today?
Are you going to upload the video onto YouTube or wait until we finish the PowerPoint?
No, I'm video of this lecture. Yes, I'll upload it. Yeah, I'll upload it right now. It'll take a while for it to process, but it'll be there.
Okay, thank you.
Yeah. And I would like to see more of you in the discussion section. Okay. All right. I'll talk to you on Wednesday, guys. Bye.
Thank you.